same time.
All right. Anyway, so let's go.
Yeah, so I think I'm up first.
Okay.
So yeah, clinic and computer division. I've transitioned to using pathogens to get more towards clinical relevancy. So in the project we just recently published back in September, which I'm showing here on the slide, we evolved 18 replicate populations of staph under increasing vancomycin concentrations. And then we sequenced the endpoint populations. So on average, it took about 9 days to evolve to vancomycin intermediate resistance, which is just a clinically defined threshold. And yeah, we sequenced the endpoint populations, and then we examined their evolutionary trade-offs to second line antibiotics. So we looked at these so-called collateral effects. And so we found through the sequencing and subsequent analyses that these 18 populations took two major evolutionary pathways as they evolved, increasing vancomycin resistance. One of the pathways, mutations occurred in IPSU, which is responsible in global stress response. And then the other pathway, mutations tended to occur in the wall-KR regulon. So this is it's involved in many phenotypes, primarily cell wall metabolism. So given this, we asked, okay, so do these divergent populations, genotypically divergent populations, how do they respond to the second line drugs? So we found that in some drugs, it actually it greatly depends upon the path that evolution took, how the populations would respond. So for example, if the population evolved mutations in IPSU, they tended to be collaterally resistant to cefazolin, whereas the populations that evolved mutations in wall-KR regulon really had no directional change one way or another relative to the ancestor. And then in nafcillin, we see significant divergence of phenotypic responses to that drug. So if the populations evolved in the IPSU pathway, they tend to be collaterally resistant, whereas in the other pathway, they tend to be collaterally sensitive. We found that when we just looked at nafcillin and just looked at which mutations within the wall-KR regulon are sufficient to cause this collateral sensitive phenotype, we found that only YCHI mutations are relevant. So these are two-component regulatory genes. Actually, coincidentally, this wall-KR regulon, there's this two-component system that regulates this pathway. It's the only essential two-component system within staph, which is interesting. So, okay, so given this divergence, right, and given the fact that there is this divergence in general, populations evolved in distinct ways to vancomycin, with sometimes contrasting phenotypic effects to second line drugs, we wanted to quantify this, the likelihood that replicate populations would trend one direction or another, collateral resistance to sensitivity. So we derived this metric called the collateral response score, which really just takes into account of the magnitude, the direction, and response, and then also the likelihood of response. And so as I'm showing here on the right-hand side of the the slide. So I'm just showing the bootstrap distributions of these collateral response scores by pathway. So we see that if the populations evolved RPS mutations are tended to be collaterally resistant, irrespective of drug relative to the regular mutations. And then in the bottom right-hand corner, this panel, where I'm stratifying both pathway and drug. So we see that in some cases, like again, nacillin, divergence in the mean response, the mean of these distributions for these two pathways, indicating yes, there is sort of a divergence. It both confirms that, but also sort of within the score itself, which I'm not showing on the slide, it allows one to easily, just by looking at the score, to see, okay, if it's positive, the score being positive, then the populations tend to be collaterally resistant. If it's negative, collaterally sensitive. And we argue that, and of course, there's more work that needs to be done. I look at this as sort of as a stepping stone towards clinical translatability. But sort of my ideal case is that we've garnered enough information, not just about staff, but whatever pathogen of interest and of relevance within a patient. We have enough information about the pathogen, both phenotypically, how it responds to drug, and genomically, that we can start to make accurate predictions of responses that is sort of encapsulated within this collateral response score. So in addition to an antibiogram, we are arguing that in the future, a collateral response score that sort of captures this likelihood is necessary for the clinician to predict where the population is going next. So that's sort of what I've been working on, and yeah.